Well, it's 9:01, so I think we should go ahead and get started. I want to thank everyone for joining our webinar today. This is the third webinar in our series of subject matter experts on the California Agricultural Neighbors Initiative process. And I hope that you've enjoyed the prior two webinars that we've hosted, and you will get additional information today out of our two presentations. I'm Norm Groot. I'm the Executive Director for Monterey County Farm Bureau and co-leader of the California Agricultural Neighbors, or CAN, process initiative. And I would like to acknowledge also on the line and in the meeting today on the panel is my co-leader of CAN, Natalie Kraut-Greenberg, who is the Director of Inspection Services Division for CDFA. Thank you, Natalie, for all your hard work that you've put in to make this happen and move forward in an expeditious manner. So I think we've accomplished a lot of great things in the last two webinars, and I look forward to not only the presentations today, but the second hour here will be our roundtable discussion where we can pose additional questions and also look back at what we learned and where we really need to go with it in the future and how we plan to expand that knowledge base based on the presentations that we have seen so far. Also want to thank California Farm Bureau Federation for hosting the meeting today, as well as Don Steckel from CDFA, who coordinated a lot of the logistics for these three webinar series, and also Tim Carden at California Farm Bureau for all the electronic logistics. So thank you all for helping putting these together. We are recording the webinar and do appreciate that ability to share those links after the webinar is completed so that others who may have missed them can view them, or we can actually go back and review for ourselves if we need to. The format today will be for the first hour, the two presentations that we have scheduled today. They will be about 20-minute presentations with a few minutes for questions afterwards. And then we will move into the roundtable discussion with our facilitator, Abby Dilley from Resolve. And thank you, Abby, for joining us today. Appreciate it very much. So we have a full agenda today, so we're going to go right into it. First of all, I wanted to go through a few logistics for everyone. We are going to use the Q&A function today for all questions from participants. So please do not use chat to ask your questions. We want to record those questions in Q&A so that we can make sure we capture all the language as well as the responses that we type back to you. So please use Q&A for all your questions, either during the presentations or during the roundtable. That means everyone's going to remain muted during the presentations, so we will use the Q&A exclusively. And if we don't get to your questions in the time allowed, we will make sure that we hold those over for further discussion with the dialogue group and also possibly during the roundtable if there's time. So why are we here today? This third webinar is going to cover on-farm factors that may affect STEC survival and contact with produce. And we have two speakers that are going to make presentations this morning. As I said, they will be about 20 minutes each, and they have slides to share, so they will be doing that. And let's move right away into our topic of discussion today, which is first presentation, the use of machine learning modeling to improve food safety quantitative microbiological risk assessments, a case study, a predictive model for survival of E. coli 0157H7 and generic E. coli in soil amended with untreated animal manure. And that will be presented by Dr. Hao Peng, who is the FDA and is a biologist and risk modeling specialist at the FDA Center for Food Safety and Applied Nutrition. His recent efforts at FDA have focused on modeling the survival of pathogens, including E. coli 0157H7 in manure amended with soils in support of the produce safety rules. So with that, I will allow Dr. Hao to begin, or Dr. Peng, excuse me, to start sharing your screen and moving into your presentation. And thank you for joining us today.
Yes, thank you very much, Norm, for the introduction. And hello, everybody. Glad to be here to present one of our recent projects on the survival of E. coli 0157H7 and Teneri E. coli in soil amended with untreated animal manure. So as Norm said, this is a case study showcase where we use machine learning to develop a predictive model to be used in the food safety risk assessment to improve our prediction. So before I start the presentation, just wanted to acknowledge my colleagues on this project, Amir Mokhtari, Yuhan Chen, David Oriang, David Ingram, Manan Sharma, Pat Mueller, and Jane Van And this is a collaboration between FDA Center for Food Safety and Applied Nutrition and USDA Agricultural Research Services. So just an outline of the topics that will be covered in this presentation. So first of all, in the produce pre-harvest environment, there are a lot of potential sources and contributing factors for microbial contamination on produce crops growing on the field. Just to name a few examples, domesticated animals, wildlife, worker health and hygiene, agricultural water, and among those factors, there is biological soil amendments of the animal origin, or they call it BSAAO, such as raw bovine manure or poultry litter. And the BSAO is one of the potential sources of microbial contamination during pre-harvest environment. So for BSAO, it is a known reservoir for enteric pathogens such as E. coli 0157H7 and Salmonella. And once BSAO is introduced into the soil through the application, they can survive in amended soil for extended times, depending on the environmental factors. And that can lead to subsequent contamination of the produce crops through different mechanisms. For example, through splash, via rainfall, or harvest. And once they reach the produce crops, they can persist through the pre-harvest environment and may pose a risk to consumers and cause an foodborne outbreak. So to that end, FDA is conducting a risk assessment to evaluate the risk of human illness associated with consumption of produce from growing areas amended with untreated BSAAO. And this information will be used to support development of a provision in the FSMA Produce Safety Rule regarding the use of BSAAO. And this risk assessment will evaluate the impact of certain intervals, especially the use of a time interval or intervals between application of the animal manure and harvest on the predicted risk to consumers. So this slide here shows a conceptual framework for the BSAO risk assessment model. So the starting point at the initial level in manure and was modeled throughout the supply chain to predict the public health risk. And as highlighted in the red box here, survival in amended soil is a key component of the risk assessment model. So there are a lot of factors that can affect pathogen survival in amended soil. And they can be broadly categorized into environmental factors and agricultural factors. For environmental factors, it can be things weather-related, for example, precipitation, temperature, and also soil moisture. And some example agricultural factors are manure type, soil management, and that application method. For example, surface application or tillage. So all these factors may have an impact on the survival of pathogens in amended soil. Okay. Okay, there we go. So the let's see. So the research objective of this project were to develop and validate a predictive model of the fate of enteric pathogens in soil amended with untreated BSAAO under dynamic environmental conditions, and to use a model to provide quantitative estimates of pathogen concentrations in amended soil over time. And also wanted to characterize the impact of influential environmental and agricultural factors on survival of enteric pathogens in amended soil, and ultimately incorporate the validated survival model into the BSAO risk assessment framework. So I would like to first start with an overview of the data we use for the model. We are very fortunate to have access to a very comprehensive data set that is the USDA manure pathogen survival study. This study on the field experiment has been published in Journal of Applied and Environmental Microbiology. This is a multi-year longitudinal field experiment conducted under different seasons and across 3 different locations in the Mid-Atlantic region. Some additional experimental conditions, including amendment type, soil management, inoculation level, and depth of manure application. So overall, the very comprehensive longitudinal study yielded 162 survival profiles each for both E. coli OFF77 and generic E. coli. And additionally, weather data were also collected, including ambient temperature and precipitation and during sampling days for each experiment field. So just to take a look at the survival profile observed from the field study, you can see that the survival patterns varies a lot. So a variety of different patterns were observed, ranging from a relatively simple exponential decline, where it started with rapid decline followed by a long tail, and all the way to more complex patterns with fluctuations between survival and growth, where they have multiple spikes throughout the survival, and with the rapid decline followed by different fluctuations. So as you can see from the figure, a total of 6 different survival patterns were identified in the field trial, and they all behave very differently. So in order to develop a model that can capture all these different survival patterns, we need a flexible approach that can accommodate these. So to that aim, we choose a machine learning algorithm for model development, and that's the random forest approach. So the random forest model described the responsive E. coli concentrations in both generic and E. coli OFF77 to changes in agricultural and environmental conditions over time. So like I showed earlier, in this field trial, a lot of metadata were collected, and the machine learning model really empowered us to include the impact of all these different factors into the model in order to predict the response of E. coli OFF7 and generic E. coli concentrations in soil amended manure over time. So just a brief introduction on the random forest approach. Random forest is a so-called ensemble method, consists of many classification and regression trees, or CART, that are created using bootstrap samples. So each CART predicts a model response based on the values assigned to the subset of explanatory variables selected in the decision tree. And the random forest combines the results from all the forest of trees to get an overall prediction for the model response. And this way it prevents overfitting, reduces variance, and provides more robust predictions. Okay, so we in the model development, we use 70% of the survival data for training the runoff forest model. And the remaining 30% were set aside and to be further used for validation of the runoff forest model. And the predictive performance of the model was evaluated by using normalized rooted mean squared error, coefficient of determination, and acceptable prediction zone. We also conduct a cross-validation based on experiment locations. So this field trial were conducted in 3 different locations across the Mid-Atlantic region. So in order to explore the model's possibility in extrapolating the model to a different geographic location, we conduct this cross-validation based on experiment locations. So each time we train the model using data from 2 of the 3 experiment locations and use the other location to test the predictive performance. Okay, so let's see some results. So we tested the performance of our developed random forest model. And these 2 figures here shows the comparison between the actual concentration observed in the survival data set on the x-axis and the predicted concentration using our model on the y-axis. Ideally, with a perfect model, all the dots will be lying on the diagonal line, and the closer the dots to the line, the better the model. So as shown in these 2 figures, you can see the model predicted well the concentration for both E. coli OFSM is 7 and genetic E. coli. You see most of the dots is surrounded near the diagonal line. And we do find statistical significant correlation between the predicted concentration and the observed concentration. And there's no significant deviation from the one-to-one line from the figure. Model predictions are satisfactory. Going to the next one. So we also compare the observed E. coli concentration in the validation data set with the predicted concentration values from the random forest model. And these 2 figures here shows the concentration observed over time. The white boxes represent the observed value, and the gray boxes represent the predicted value. So you can see overall they are in pretty good agreement. The predicted median, the interquartile values are in good agreement with the observed value. And from the statistical measures, the normalized rooted mean squared error is 5.2%, R squared is 0.9%, and 93% is in the acceptable prediction zone. So pretty good results overall. And another thing we wanted to test the model is its ability to capture different survival patterns. As I shown earlier in the presentation, there are many different survival patterns observed in the survival data set, and many of those are very complex with spikes and non-monotonic features. So do want to see if the model can capture all these different survival patterns. So in these figures, the dots represent the actual observation from the survival data set, and the solid line represents the model's prediction, and the dotted line represents the prediction range. So we can see for all different survival patterns, the random forest model prediction can capture these different survival patterns very well. So it can capture the spikes, the up and down, the fluctuation between survival and growth for different patterns. Most of the adults are falling within the prediction range. That indicates our model were able to capture the complex and non-monotonic relationships often observed in the survival profiles over time. So for the cross-validation based on experiment locations, each time we use data from 2 of the 3 locations and trying to predict the other location. So you can see the statistical measure here. So overall, in these cross-validation, the prediction performance were less accurate compared to the model trained using the whole data set. And that really emphasized the importance of data representativeness and the size of data in developing a machine learning model to provide accurate prediction. And also the reason for one of the locations, like the University of Maryland Eastern Shore locations, the results were less accurate is probably due to the difference in the data set. So for the other 2 locations, experiments were conducted in 3 seasons, and in this UMES location, the experiment was conducted in all 4 seasons. So when you train the model using only part of the data and try to predict the survival in all 4 different seasons, the results will be less accurate. So like I said, that emphasized the importance of data representativeness. We also used our developed random forest model to rank the relative importance of different factors. So this figure here shows the list of explanatory variables ranked based on their relative magnitude of importance using permutation importance score as a measure. So the higher the score, the bigger the influence. So from the model, the most influential factors are base post-inoculation, inoculation level, amendment type, and for environmental factors, rainfall and soil moisture content play an important role in the prediction of survival of pathogen in soil amended with animal manure. We also evaluated how those influential variables may impact the predicted E. coli concentration in amended soil. So here you see how E. coli concentration is impacted by different factors. So on top, we have the impact of days post-inoculation and the inoculation level. So you can see in general, as days goes on, the overall concentration goes down. And not surprisingly, the higher the inoculation level and the higher the concentration in soil amended manure. But for environmental factors, you can see in general, higher precipitation and higher soil moisture is associated with higher concentration in soil amended manure. And for amendment type and E. coli type, there's also a difference in concentration. But another finding we can observe from these figures is that in general, generic E. coli and E. coli 0157H7 in the maiden soil seems to be a decline over time at a similar rate. So you can see the 2 lines are almost parallel in different figures, and that also indicates the impact of agricultural and environmental variables is similar between E. coli and E. coli 0157H7. And we actually can use the model we developed for E. coli 0157 to predict the generic E. coli concentration pretty well, and vice versa. So that indicates it may be possible to use generic E. coli as a surrogate to investigate the impact of different variables on the survival of E. coli 0157H7. So in conclusion, in this study, we were able to develop and validate a predictive survival model for E. coli 0157H7 and genetic E. coli in amended soil with the SAAO. The developed random forest model captured the distinct E. coli survival patterns over time, especially those non-monotonic and fluctuations in pathogen population over time. And it accurately predicted changes in concentrations of E. coli in amended soil under dynamic agricultural and environmental conditions. And also, we use the model to identify the influential factors and characterize their impact on the E. coli concentration in amended soil. In the modeling framework presented can be used for similar pathogens such as Salmonella when similar data elements become available. Also, from the cross-validation, it indicates the model can be potentially generalized to other geographic regions. The representativeness and size of the training data set were crucial, impacting the model's prediction accuracy. So that means data sets that contain information from additional geographic regions, when available, can be used to validate the random forest model or to be incorporated into the model to enhance the model's predictive performance. So finally, I wanted to acknowledge Dr. Brian Van from USDA ARS for statistical consultation, Dr. Fauzi Hashim from UMES for laboratory and field research contributions, and many, many others from USDA ARS for their field and lab technical assistance. And also this research was supported in part by ORISE and also University of Maryland, FDA, Joint Institute for Food Safety and Applied Nutrition through a cooperative agreement. And the project presented has been published on the Risk Analysis Journal. And with that, I wanted to conclude my presentation and open to any questions.
All right, thank you, Dr. Peng. Appreciate that very much. And I'll remind all the participants to please put your questions in the Q&A function, if you could, please. We do have one question that came in on your slide number 17. If you could back up to that real quick. You're showing a decline over time in the various conditions there. Just wondering how that plot might be represented with our Salinas Valley conditions here. And given the type of intensive agriculture that we have in the Salinas Valley, and how long before the STEC might go from starting to get conditions in manure as it's applied to the levels that minimize risk? That's a lot of question there. So just wondering if there's a correlation to what we're doing here in the Salinas Valley.
Well, for this model in particular, all the data we used was based on experiment in the East Coast, so namely the Mid-Atlantic region. Although many environmental factors were collected, including temperature, precipitation, and soil moisture, there could be other factors that also impact the survival, and that can be very different from Salinas Valley and the East Coast in the Mid-Atlantic region. So it's hard to say how well this can reflect the situation on the West Coast, especially in the Salinas Valley, because of many of the unique environmental factors and also the agricultural practice. So it's really hard to say without any data from that region as an input to the model. So as data become more and more available from that region, that can be incorporated into the model so we can have a better understanding on that. Is there another part of the question that
Yeah,
I'm
there's
not
another
sure.
question now relating to soil type and whether or not that makes a difference.
Soil type.
Yes.
Well, in this particular study, soil type was not included as a factor. The only factor related to soil is well, there are several. One is soil moisture, but also the soil management. And in this case, it's whether it's managed organically or conventionally. But soil type was not included in the model because the data that we have does not include that as a variable.
Okay, thank you. And then another maybe last question here. Was there an observation between the application time and the planting date, so to speak, of a crop? And does that make a difference if it's longer or shorter?
Application time and planting time?
The application, yeah.
I don't think, but that may be presented somewhere in the paper where it described the original study design and the approach you're following in the longitudinal field study. In this study here, we focused on developing the model using their data for prediction. So I can't really give a good answer to that, but that information might be in the Manan Sharma paper that I presented earlier in the presentation that described the study design.
Okay, great. Well, thank you for taking a few questions here, and thank you very much for your presentation. Great information here, and hopefully everyone will be able to access that either through your report or when they view the video link later on. So thank you, and we'll move on to our next speaker. If you could take down your presentation. Our next and second speaker is Dr. Trevor Suslow on STEC in leafy greens, always there or a seasonal arrival. Dr. Suslow is an emeritus extension research specialist at UC Davis and is well known for his long career working with California specialty crops, ranging from row crops to vines to tree crops. He has spent decades involved with diverse leafy greens in particular, and he continues to interact across trade associations and consult for the produce supply chain on safety and quality issues with substantial efforts in the Salinas growing area. Thank you and welcome, Dr. Suslow. You can start your presentation.
All right, so in any case, what happened along the way is I was asked to cover production patterns and practices. And I can certainly work in the other aspects. I also mentioned I just presented a lot of that information yesterday with CPS. That recording will be up there. So it's a little odd to be in the position to talk about this rather than have somebody from industry, but maybe I'm the neutral party here. And there is an updated version that covers a lot of this. That is part of the California Ag Neighbors effort. I was asked not to really use that. And so, everything that's old is new again. I pulled up, parts of a presentation since there's relatively limited time that's really adapted from, 2006, 2011 presentation, that I presented several times, developed by myself, Belinda Platt, for those that remember her, and Mary Ziske. So let's go ahead and get into it. For those that maybe aren't as familiar, just to get a sense of the industry and the locations that we're really referring to, largely focused on this more northern Central Coast area that includes some more interior areas, even as I'll touch on, historically a much more common production out in that Fresno County area. It seems to be, swings back and forth a little bit, but also into, Santa Clara a bit and down in Ventura. Then, of course, during the winter seasons, which, are still very relevant in Imperial County and over into the Yuma area, which often gets lumped together. I started to, and they said, what the heck, it hasn't changed so much. The key aspect of all of this is that between California and Arizona, over 98% of the US production of leaf lettuces across the board happens in California and Arizona. You can kind of see, at least in 2009, from those statistics, the distribution from head, romaine, leaf lettuce, and spinach. And it varies, and there's ups and downs across. That would be nice if I could advance. There we go. Just briefly, the most recent statistics available, 2019, you can kind of see what that distribution of production in terms of weight, which is the one that was available, how that distributes in the USDA and statistics. This out of the way. But part of the things, again, drawing on the data that's available, really looking at per capita consumption. Here you can see it over a pretty long timeline. You can see that there's ups and downs, but in general, for the head or iceberg type of lettuce, per capita consumption has kind of stabilized around that average level with differences across the years. And then romaine and leaf lettuce in this databases are lumped together, and you can see sort of as romaine and others have come up, maybe iceberg has come down. A lot of others that have participated in this Ag Neighbors dialogue groups have much more granular details related to this. But I wanted to give a sense of what was happening, at least across from production to consumer consumption. So what I'm going to go through now is to just walk through some information that relates to the timelines for growing and harvesting activities in each growing region, a bit of comparison of the activities and the pattern of growing activities, really the rotations that may be encountered within a single firm operation or a single ranch area. And then some of the comparisons that we have to keep in mind for the growing regions, and actually took out, for time's sake, the irrigation practices and a lot of the images around what all these actual practices look like. But they're available, and we can always share those out in a different way. I mean, one of the key things here is that sustainability and really profitability requires in many of the areas very quick turns from crop to crop, and that is really, I think, relevant to the prior presentation. Although manure use, non-composted manure use, perhaps may not be a relevant factor per se, but I'll share some data with you from actual on-farm studies, with other ways to look at survival of these pathogens or pathogen surrogates in soil. So, for the different regions, sort of lumping Salinas and Santa Maria production activities together from planting, growing, harvesting, and ground prep, the colored bars against calendar year are for iceberg lettuce. The leaf lettuces, romaine, is somewhat different. You can read the caption there, and they can go out through say the early part of November. Again, you would have to separate out specific Salinas area, some of the Santa Maria and other production regions, and even the microclimates and positions. And this is really important aspect to keep in mind as one starts to think about prevention, mitigation, and understanding some of the causes around the patterns of outbreaks. I mean, it's very important to recognize that just because there's, say, romaine lettuce coming from the Salinas region in July, that doesn't mean it's coming from everywhere across that broad valley. And in the more southern regions where the temperatures are really high, if you have cases that start to develop, a little bit after that, the product is highly unlikely, if not, not possible, to have come from that region. So you have to keep all of those pieces of the puzzle in mind when you evaluate what's going on and what might explain some of the STEC and 0157 presence and relationship with illness and outbreaks. This Huron gap that I mentioned over sort of in the Fresno County, it was, much more involved in sort of that small window, in between the north to south, south to north movement. It's much more limited. But again, one hears at least that, there is a little bit more movement back there to fill in some time slots for product availability, leaving the desert earlier or, coming back a little bit different. So those two planting to harvest activities are kind of split around that north to south, south to north transition in that fashion. Then again, moving down into the winter desert region, you can see the timeline again is for iceberg. And what you're really doing again is a lot of that ground prep, is happening during the summer months, preparation, planting, growing, and harvesting, and then it continues on into the following year, the following season. So I'm going to move into then some production patterns on individual ranches. And, it's important to keep in mind that multiple lots are in production simultaneously. Different lots can be in production, often for different crops, immediately adjacent to each other. And all of these activities, planting, growing, harvesting, can occur simultaneously on the same ranch block. And of course, they can be geographically diverse over any given region. So here would be an example. Again, in all of these things, there's details, there's exceptions, but this is sort of the higher level. On average, these things, are fairly typical. So one would be a lettuce to lettuce rotation in lot 1. Lot 2 may be coming out of strawberries and go back into some leafy green. Lot 3, would be in broccoli as a rotation in between from previous year, and then getting into lettuce for a later season harvest. And others that again is, lettuce to lettuce of different types, in the back-to-back rotation. So all of these things are possible, and it depends on the specific management and what that firm has in mind for a piece of ground. So trying to look at some of the other factors that are relevant to keep in mind. In general, the number of crops per land acre per year in that sort of central coast, north and south, Salinas to Santa Maria, 2 to maybe 3 on the same piece of ground in consecutive years. And if you look at another factor, which is important to keep in mind, is like the duration of development. This is for, again, focused on head lettuce. This is how it was, this was organized at the time. So you can see early in the season, cooler conditions, longer days to maturity, mid-season, the fastest, and then a little bit later, in that relevant season, it takes a little bit longer. And you can see that across that sort of, Central Valley, San Joaquin Central Valley area, and into Imperial. But here, one of the key factors with Imperial and Yuma, at least, you're really looking at probably one crop cycle within that particular seasonal year. So, one of the things that we've considered for a long time, which I think is very relevant to, some of the questions about, within that die-off of, a soil amendment applied, as you heard in the previous talk, when does that long survival tail reach a level where it no longer is of significant risk? And it's been something that's been tried to be predicted for a very long time, and it's so variable it's hard to come up with it. But one of them is, is the crop sequence and the season, something we could target. The crop residue, the speed of the crop cycle following, any type of amendment, including the crop itself, and is this a site for soil amplification? So one of the things that's related to that, it wasn't clear from the models. It seemed like that at least ambient temperature on the East Coast wasn't a significant factor or as significant factor in the rate of die-off. But here, you're really talking about as you move across the different production patterns, cool to warm in the Salinas area, and then moving as you get out of the mid-season from warm to cool. Whereas in Imperial and Yuma, you're going, say, from hot conditions, right ahead of planting, to colder and then cold into warmer as it stretches out. And again, a number of the things have changed over the years, characterized by at least not a lot of rainfall, depending on the year here in California, but certainly more than in the other areas. But one of the other things I was asked to touch upon that I think will lead into the roundtable is something. What do we know about die-off? And there are a few other studies. I think these perhaps are two that one might turn to, and really just trying to summarize the basic conclusions. In these studies, most die-off, when you're applying it to try and simulate irrigation contamination, overhead irrigation contamination, the bulk of the die-off happens really in the first 3 days with the available strains and all the other caveats one could throw at it. In some cases, 1.5 days, you have most of the highest level of death, high heat, low relative humidity, high UV exposure. It can even be less than a day, but you always have pockets of survivors if you look hard enough and if you sample broadly enough to 21 days. And that's what the lower paper really addressed. And these were done in an open environment. The one in the bottom half there was done down in the Salinas region out on commercial farmland. So this is pretty typical of what we see here. I'm showing you applied, through a sprinkler system, the research farm, but whoops, in the Salinas area, unrealistically high application that ended up in the soil. But you can see at least 30 days out to about the limit of detection. If you do an enrichment and test by PCR, you can track that out to about 45 days. So that's through water. This is what I think perhaps is a little bit more relevant. A number of studies where we looked at adding some of the things, replicating what we found from talking with growers. When it's applied to the soil in an organic carrier that has some nutrients or as a spray on, we always see this spike of application in the first few days, in soil. And then again, as you saw in many graphs, this sort of slow decline and long survival tail. So here with this type of approach to a contamination of soil itself, again, about 45 days through enrichment, you could get culturally recoverable isolates following an enrichment. So they've dropped really low, but they're still there and they're going to be there in pockets. And the same thing with if a crop was contaminated by some level, even if it's not something that would result in an outbreak level, when you incorporate those crop residues into the soil, we see the same thing. We see that initial spike here, much lower level was used sort of for early incorporation, but very similar, if that crop, in this case, spinach, went to maturity. And again, you can see these are done out in an open field environment in a research dedicated commercial field that was made available to the group at the time. So the final area that I was asked to touch on, to just give some background, because it apparently was a question that has come up in previous sessions a few times. This is the study that remains one of the few to explore some of the underlying causes for variability in bacterial community composition as a variety of, as a function of variety, time, and environment. So this was all done, several years ago, funded by the Center for Produce Safety. And you can see the areas that it covered. So, I went out and collected romaine from a number of fields in transects over time, both in the Central Coast area and down in the desert at the appropriate time. And I'm just going to touch on some very quick, high level, takeaways. And really, one of the things that was found is that samples from the same field in terms of the microbial community, the bacterial microbial community, composition were much more similar to each other than other samples taken as you moved geographically down the valley, even within the same day. So that's one. But I think one of the key findings from this effort, and I've greatly simplified this graph quite a bit, is that you see a very distinct difference in the bacterial community composition between romaine lettuce that was harvested during the summer production cycle versus the desert winter region. So, there's very different conditions. Variety didn't seem to play a major role, but that wasn't explored heavily. There wasn't a lot of diversity in the romaine that was available for us to sample. But this was a pretty stark, I think, difference that happened. Then the follow-up studies really demonstrated that planting two different types of leafy greens, romaine lettuce and spinach, in the same field and then sampled on the same day. And if you simply look at the colors, you can see that very different composition of the bacterial community develops, even though they're exposed to really the same conditions. So variety, plant, physiology, whatever it is, definitely makes a difference in what's going to be there. A different study looking at somewhat the same thing is that leafy greens really develop different bacterial communities within the same field. Again, looking at head lettuce, romaine, and baby spinach, simply represented by these overlapping colored fields. They really have a different bacterial microbial community composition that develops on them due to the contribution of the plant. And one of the things that was, I think, very interesting, in terms of how we approach this, is that the plant-associated bacteria on the surface of the leaves are less diverse and far more culturable. So there's this culture-independent approach. Things, we know that there's many things, especially in the soil, that you simply can't easily culture out everything that's there. It's extremely diverse. But on the plants, it doesn't seem to work that way. You have a very high correlation between what you can culture and then what you can find by simply looking for their DNA signatures. So again, a lot of busy stuff going on. But I'm just saying there is this information. There are other studies underway. There are other studies being reported, but this is very specific for the California situation. So it can change a little bit about how much emphasis we put on trying to have these culture independent methods, unless there's a reason to do that. Then the last, to wrap it up, I was asked to just comment briefly on what quantity of STEC contamination causes an outbreak. And I don't think we have a hard answer, but I just suggest that if you want to look at the resources posted on the United Fresh website. This is the title of it. Would my sampling plan have detected contamination levels that resulted in an outbreak? It's really a thought experiment. I'll walk through a couple pieces of that. And then some very detailed, very elaborate work recently presented. There's a final report on the Center for Produce Safety website done by Matthew Stasowitz and his collaborators on simulating, again, field-based, risk-based sampling, trying to say, predict, what you needed to detect and how that related to public health risk and outbreak. So I guess I would suggest that sort of the best available pre-harvest scenario boils down to approximately 1 colony forming unit, or 1 cell per pound of product. And the foundation for this estimate, with a number of assumptions that one has to take, this thought experiment, and others have sort of triangulated really to the same point, is based on information provided by FDA from a 2018 outbreak of romaine lettuce. All of this information is posted at United Fresh. So 45 acres, 62 clinical cases that were known, using a CDC-based multiplier of 26, about 1,600. Taking sort of a typical yield per acre of romaine and estimated number of servings per harvested head. This is where that number comes from. So that was a lot of different things without getting into, this question of, is, the recurring 015787, always around or continually reintroduced, or is there some, reservoir that seasonally amplifies due to the conditions, and I don't think we have a sound answer, but I think it's really a little bit of all the above. So with that, thank you for your attention, and I'll turn it back to Norman, and I apologize for the mishaps with technologies, and we can get into some Q&A.
All right, thank you, Trevor. Appreciate the presentation. And I'll start with a question from one of our participants. Given your work and Dr. Cooper from Arizona with low mutation, what's needed to figure out if we have reservoirs of pathogens or reintroductions and amplifications? What do we need to explore next?
Well, I think the quick answer is really, I think we need to continue to explore the opportunities within this working group, within this collection of industries brought together to talk about, sort of regionally based shared solutions to all of this. And, I'll argue for quite some time, it's hard for me to answer some of it. I'll probably lean on some of the other folks in the roundtable because there are some understandable confidentiality barriers to sharing what exactly is going on, say in the longitudinal study, what is going on in some other areas. But it's really access to be able to experimentally ask the questions and look at the different potential sources. I think it's clear that they're there. I think some of the emerging information from those that are looking at evolutionary genomics of 015787, as I said yesterday, for those that might have been on that symposium webinar, we shouldn't be so surprised that you don't see a lot of mutation or a lot of diversification among these strains. But I think one of the key questions that we need to try and answer is partly those people with the expertise to really do some deep mining of the sequence and genomic information available across that set of isolates to look for those factors which provide environmental fitness, which provide a competitive advantage and start to challenge that. I think it's a little bit, of all of the above, as I've said, but it's hard to get at that because of the concern and sensitivities, which I think are very understandable around the industry, and allowing that to happen, to discover something that would strongly implicate, them, a piece of ground or a whole region. So it's not trivial.
Okay, and then related question, has there been any evidence that soils from the Salinas Valley area are reservoirs of E. coli?
Great question. And I'll try and answer it in two ways. One, in my experience, we don't have evidence that is the case, over time, unless you as we did, from a research standpoint, you sort of construct, the opportunity to see long survival. Less so for salmonella, I'll just throw that in. And we've done, rapid response projects, and clearly, they can be part of sort of a stealth reservoir in soil. Others that I don't feel it's my place to share their data have been looking for evidence of that. And, really, I think the conclusion is, if it's happening, it's in, it's hidden in a way that any type of sampling regime, would not seem to find it and make a strong case for that being the reservoir to focus on.
Okay, great. Real quick, one last question here. Does plant population have a factor on survival of pathogens?
Good question again. I don't have an answer for that, other than certainly from I think our work, stage of growth, I think, and stage of plant development and its architecture, I think definitely has plays a role in the likelihood of survivability, should contamination come to that crop.
Okay, great. Well, thank you for answering questions, and very much thank you for your presentation, Dr. Suslow. Great information there, and hopefully everyone can use the video link later on to review all those great graphs that you presented to us. At this point, we're going to move into the roundtable discussion. I'm going to turn the meeting over to our facilitator, Abby Dilley, with Resolve. Go ahead, Abby.
Thanks very much, Norm. So this portion of the webinar today is to continue benefiting from our interaction with our subject matter experts, both the panelists today, but over the course, also including panelists and subject matter experts from our 2 previous webinar sessions, so the full series, and to have the benefit of being able to ask them questions, and both some of the questions that were submitted during those webinars as well as today, and then also any other questions that people submit in the Q&A segment over the course of the conversation for the next hour. Additionally, it's really representing a pivot towards taking all the information, the cumulative information and insights from the webinar series and start processing that information for the dialogue group to pick up that information and their deliberations and sharpen the focus that was the interim report, represented the interim report, and ask the question to have the conversation among the dialogue group about what does this all mean to help guide where we want to focus for options for action steps that will ultimately be represented in a final report. And so we have the agenda as laid out is to do both of those things. We'll first turn to Tim York from Leafy Green Marketing Agreement, Jack Rice with the California Cattlemen's Association, and Jim Gorney with FDA to give their reflections on what they've heard with some specific focus on different dimensions of the webinar series. And then we'll open it up to the larger group and panelists to talk about through how are we processing this information and what have we gleaned insight-wise from the webinar series. And then what should the dialogue group, really looking to all of you to give the dialogue group some guidance in terms of where to focus their deliberations to take in this information and sort through and prioritize options for action steps. And that's also looking for some guidance, not only on the content of the interim report and some of the information that's been gathered, but also where there still are outstanding questions. We have some flexibility in the work plan to be able to potentially probe some additional information, but just really want to keep in mind that the objective of the CAN group, the California Ag Neighbors group, is to land on a set of action steps that will be contained in a final report. That's the next iteration of the interim report released recently. And also some process advice. So again, some potential for circling back to others. Then we'll go back specifically to submitted questions and pick up and engage with our panelists from all of the webinar sessions to pick up those questions and gather additional information and really maximize the value of having our subject matter experts here to follow up with those questions. And then Norm, I'll hand it back to Norm to talk about the immediate next steps of where we go from concluding the webinar series and then turning back into our dialogue group for further deliberation. So that is the layout of the agenda for this particular hour. I'm going to turn immediately to Tim York to kick us off as a discussion starter, to just ask your insights from what you're taking away from the webinar series and guidance you may give to the dialogue group to sharpen their focus on their deliberations going forward. So Tim, to you.
Great, thank you, Abby, and good morning to everyone. As you may know, the LGMA produces about 99% of the lettuce and leafy greens grown in the state of California are produced by LGMA members. And so our members have to follow the standards that we develop. I'm really struck in these 3 webinars in particular, again, how complex the issues really are that we're dealing with, from the seasonality to the planting to the harvesting and all the various factors within there. When you look at this slides that Trevor showed with crops that are in the ground for over 100 days and exposed to elements and whatever else they may suffer, how complex this issue really is. And yet the FDA report that was released on April the 6th, really said to us, it may be complex, but you need to fix it anyway. There was a clear message to the leafy greens industry that this recurring strain, recurring region, recurring issues needs to stop. And that certainly has been a message heard loud and clear within our industry. You know, the CDFA put together, I think, a great roadmap that demonstrated the direction that they would be taking in helping the industry in terms of pathogens and food safety. And that was really 3 different areas: look at knowledge gaps, and then prevention and response. And they actually put the Ag Neighbors Project under the prevention aspect, but it's really about filling knowledge gaps also. And this is something that, if you've been part of these discussions these last months, as you listen to the wine grape growers or experts from within the cattle industry or the produce industry, the knowledge gaps amongst those parties, that they had really a lack of knowledge amongst each other. And so this has really been a great response, this whole process, in terms of sharing knowledge and best practices. You know, what I'm most struck by, Abby and others, is the need for collaboration in this whole aspect. Deputy Commissioner Frank Yanis called it out in his comments about the FDA report, said, you know, FDA cannot solve this on their own, and it takes industry leadership and collaboration. And I certainly think that between Farm Bureau and CDFA, putting this series together has been a great look at what industry leadership can accomplish. So this has been a great forum for this. You know, the next steps as laid out in the report show that, you know, we were in an information share, and then we're in that stage of querying experts and subject matter experts. And then the next step would be to determine better practices. So we all have best practices in place, right? We're within LGMA, we're launching very shortly new best practices around adjacent land use and about soil inputs use. But that's based on the knowledge that we have today. And so this process needs to continue, the collaboration and interaction with subject matter experts to help inform how we can make those even better in the future. We're proud of the work that we've done to date, but know that lots of work remains ahead of us. And thank you for the opportunity to participate in this forum and bring all these experts together. It's been very valuable from our standpoint. So thank you.
Thanks very much, Tim. I think you echoed something that Dr. Suslow said in terms of, you know, it's a very complex puzzle, and we have to hold that all in our heads as we're trying to highlight better practices. And that's no small task, but hopefully with a series of webinars that will help sharpen how best to hold those pieces in our hands to come up with some specific options and action steps. So now I want to turn to Jack Rice, and I've been informed that Jim Gorney wasn't able to join us today, so gives us a little bit more time, Jack, so that you don't feel too pressured, and then we'll open it up for broader discussion. And as Norm noted, we're asking everyone to participate in that using the Q&A function. So without further comment, I'll hand it over to Jack Rice. Thank you.
Thank you, Abby. Appreciate it. The 3 seminars have been fascinating. And the first thing on my list was the same as Tim's, which is the complexity of it. It's just incredibly complicated. And I knew that, but I don't think I fully appreciated just how every facet seems to have a host of internal variables and uncertainties that go with it. It seems like, and this is probably new, because I'm newer to this process, but even the name, you've got pathogenic E. coli, O157H7, and STEC, and it seems like all those are kind of talking about the same thing, but they're all also a little different. It just struck me how complicated this is. Another thing that I really learned is that this preventing contamination seems to be really about reducing the odds. You know, it's a statistics issue. You're not going to eliminate O157H7 from the environment entirely, but there's things you do to shift those odds. You know, looking at how it moved around, obviously cattlemen's looking at the livestock and the animal side of it, the wildlife. You see that it can be carried by all kinds of animals. It can potentially contaminate from any of those animals, can result in contamination in a number of ways. It seems like there's a limitless number of paths something can happen that none of us want to see happen. To the extent that's right, this makes the discussions about solutions and next steps harder, longer, it probably just shows how important this conversation is. Just because it's one of the issues is you can do a lot of things that'll shift those odds, but it doesn't necessarily, from what I understand, mean they're all worth doing. You have to draw the line, you know, what's worth doing and what is not worth doing, even if they push the odds the same way. But this process has certainly accomplished a lot in the knowledge sharing. I think one of the most important issues has been highlighted is that there are getting the neighbors to talk to each other is a key step, needs to continue, and that's happening here. Another thing that I've learned, and I think it was promoted by this knowledge sharing, is avoiding leaping to conclusions. There's a lot of pressure to act and to act quickly. With complex problems, it takes a lot of work to avoid jumping to whatever seems like the easiest conclusion. Availability bias pulls us in these complex situations with a lot of pressure, but this helps us avoid that and work through it in a better means. Where some more focus is needed, I think continuing the conversation is really key. Maybe focusing in on just how those conversations happen, what neighbors say, what are they going to talk about, what are the ideas that are out there, and how can that be constructive and bilateral as opposed to unilateral. Another thing is just understanding more about when you shift those odds, how do you change other odds? Using the thing that is of concern to a lot of cattle ranchers, if you exclude cattle from a given area, and that reduces the chances that livestock, cattle, could result in contamination, but might that increase the odds that wildlife can serve the same function? And how do those relate, and how do you grapple with and balance those goals? So all in all, it's been great. I appreciated being part of it, and I think it has started us down a key path, or rather moved us further down a key path. Thank you.
Thanks very much, Jack. I think a good challenge to prioritize actions, and not necessarily the easiest, jump on the easiest, but try and sort through for the dialogue group to sort through where can you maximize the benefit on some actions versus others, and including more guidance on those things that have started within the dialogue group in terms of a lot more communication, neighbor to neighbor, to help address the issue. So we do want to take our time now to just open it up to the larger group, and it's an opportunity to get additional insights from the series of webinars, both in terms of how it may make you think about the interim reports, preliminary ideas and content, and where to maybe sharpen the discussions for the group to, in fact, do what Jack and Tim suggested in holding this complex puzzle and try to prioritize our areas of focus and process the information we've gathered. Also looking for where some big major gaps that remain that we maybe need to focus on in the near term to help supplement our thinking. And then lastly, any process suggestions. So again, please use the Q&A box to add your comments, but we're looking from the full group and panelists are certainly able to raise your hand and chime in, and participants use the Q&A function to stimulate that dialogue. It's really, what all of you learned from the webinar that you think really should be highlighted for the California Ag Neighbors Dialogue Group to really take into consideration, and any process suggestions going forward.
So I'll kick off the discussion here and get things going, and our panelists can jump in on this. And we've had a lot of discussion about weather and climate change, and we had some great presentations from Dr. Pathak and Parker on the seasonality and how that might impact climate conditions in, excuse me, the Central Coast region here. And then, Scott Violini gave us his take on production practices for rangelands and his cattle operation. And we just heard from Dr. Suslow some of the leafy greens statistics. And, you know, and even today, Dr. Pang indicated some environmental conditions through his model on STEC survival. So I'm just wondering if our panelists would like to address, based on what we've heard through these presentations, how can weather and climate really impact food safety assessments at this point? And should that be really a key factor in moving forward?
Norm.
Yeah, go ahead, Scott.
Scott Violini. If I could address that question, the wind always blows in the Salinas Valley. It just seems never to stop. A case this morning, you know, I'm at 1,500 feet here on the backside of Mount Toro, but it was so foggy here this morning. It was wet. I mean, I had to put a coat on just to stay dry. The weather just cleared out, and it's already 70 to 75 degrees. I don't know if this really answers the question, but our climate here in the Salinas Valley is, I mean, it's so up and down. It's crazy. Like I said, the wind's always blowing in South County, and yeah. So.
All right, thank you for that, Scott. Any other panelists like to respond to that or offer their observations on weather and climate change? All right, as a follow-up then, I guess if we're talking wind, what does that mean for the spread of pathogens and particularly E. coli?
Have any studies, I assume I heard there were some studies done on generic E. coli and the you know, how far they travel, similar to kind of the same results I used to hear about when they were checking for methane and ammonia coming out of a dairy. They set up these little screens at so many feet. And, you know, with our livestock movement, you know, we always have to consider that we have buffer zones, a minimum, I believe, of 800 feet. Since I'm not a Scientologist or a, you know, that way, I mean, dust travels, but we always have to take in consideration that we do have an 800-foot buffer zone, and a majority of the valley, we're parallel, I believe, in our production. We're running parallel to one another as opposed to north to south, because the wind always blows east to west. No, it always blows north to south. That's right.
Right.
So maybe I can weigh in with a little bit of background, you know, on this topic, you know, and, you know, the answer is certainly that is an area of concern, an area of interest as far as a risk factor or contributor to, you know, risk potential for contamination. Just to try and give a short answer, which is often very difficult for me, you know, we did a number of studies, you know, early on looking at, you know, both methods development, but, you know, and sort of more simplistic methods development, as well as, you know, looking at these distances. And, you know, what it really came down to is, yeah, if you create the dust storm, or if you create the, you know, water droplet aerosol plume, you can easily track what's moving downwind, you know, to, you know, 400, 500, 600 feet. The big difference was, you know, dispersal is a very different thing, you know, to study, and it's been studied, you know, a lot, and there are a lot of models looking at different particulate movement, different, you know, things, you know, including bacterial movement, that have been done, but deposition onto the plant and predicting survival, you know, with the techniques available has been, you know, much more complicated, and it's been done within plant pathology for plant pathogens a lot, and some other things. So, you know, position, upwind, downwind, crosswind, wind speed, all of those things matter. Things survive better, move farther during the night than they do during the day. Again, there are publications that provide that data and lay out some of those models. But dilution, the dilution factor is extreme. So you have hyperlocal, local, short distance, and then long distance movement and deposition that certainly can happen. And I'll just share briefly. Really had one real world opportunity to take a look at this in a commercial field where E. coli 15787 was detected. That particular part of the production block, a fairly large block, was not harvested, disc down. And this was downwind from a relatively small corral that had various animals moving in and out. This is in the Salinas area. But I think the most interesting thing, which I think tells us something about what we have to keep in mind and how we would look at this, is that pretty intensive sampling across, this was romaine, in a grid fashion across the remaining block demonstrated that detection wasn't found, at that setback edge. All the positives were more central. The other piece of that I just want to lay out for people to consider is that while we have the isolates from that romaine lettuce, 015787, you never get permission to go into the animal feeding operation to find out whether that was the source, to match them genetically. So until we can put these pieces together, we'll always have that open question. It's like, okay, well, you found it, but it was a bird, it was, whatever, other thing other than it came from that feedlot surface material. So, definitely, Scott, it's an important question to answer. It is related. When we look, the time of day we look, and where we look is going to be critical.
Can I address that question real quick? Thank you, Dr. Suslow. And I know you've been studying.
Trevor works best, and you have my permission.
Okay. Hey, I know you've been working on this stuff since 06. I mean, I remember when I first met
before that.
Well, yeah. But here, you've been hanging out with us. We had some of a discussion on the latter part of your comment. It's real hard for me as an operator to say, yeah. Here's the thing. I think a lot of the livestock industry is scared to death to the fact that it's government, and how do we maintain neutrality to where we don't get sued if we allow somebody to come on the property to keep up with the studies and come on. We had a discussion the other day with a couple of guys, and that's their biggest fear is being sued by the general public. I mean, that it could come back and really bite them in the butt. I mean, is there any possible way that you know of? I know I'm putting you on the spot.
Okay.
So I've heard that, like Dr. Rob Atwell, he said, hey, we designate them as A, B, C, D, E, and nobody ever knows, and it's all good.
Trevor, do you want to respond to that quickly, and then we'll go to some of the Q&A comments, and then back to some additional questions.
Sure.
Trevor,
well, I think, watch, this is going to be really short. I think we have to develop a process that allows for that to happen. I absolutely understand the concerns, but at least in our minds, getting the data, knowing the answer gives you the opportunity to do something about it. And that's really what it comes down to. So, I think at least the current data that's being developed would strongly indicate that in a cow-calf, stocker cattle, grazing pastured area, that is probably not the most likely point of focus for being problematic, because it's very, very different than a CAFO, and you're not really creating the aerosols that would likely contribute to that type of contamination. Now, we have to get there to the end point, but at least that's where it's looking. But if we don't ask the question experimentally, we can't work towards prevention. Thanks, Abby.
Thank you, and I'm glad that you're still around.
Thanks. I'm going to pick up just some of the questions in the Q&A and encourage people. Again, the goal is to collect more information, insights, what people have learned from the webinar series to highlight for the dialogue group. And then I'll turn back to Norm here shortly for some additional question roundup, because we're now merging into that portion of the agenda. But a couple things. One is from John Massa, highlighted, I believe, some areas that you think were not included in the webinar series or in significant detail. One is filth flies, one is human factors or other human factors along the biosecurity realm, but other situations such as the example you gave was homeless camps along the Salinas River, and then also plant populations. So you highlighted those three areas. And so again, that goes to the question from the group in terms of what are we missing? Do we need to engage with other subject matter experts on other parts of the puzzle? And then, and forgive me if I mispronounce your name, but Deborah McElhaney highlighted just how complex this picture is, and that currently the state of affairs, it feels like the cattle are the sole focus as opposed to the complexity of the full picture, and jumping to a conclusion that vaccination is the answer. And I think for the dialogue group, and this is echoed from the comments from Tim and Jack, is that just how complex this is, that part of maybe the final report needs to lay out that complexity a little bit more and how we've benefited from the webinar series. And that also that we need to go through this process of looking at some of the different pieces and their relative benefit. And that includes not just, it may include vaccinations as one piece of it. I don't think, Michelle J. Russell may be able to confirm that study is underway and won't be concluded by the end of the CAN dialogue or our timeline. That's one possibility, but there are many other possibilities that not only the group has identified even before the webinar series, but also in addition to the webinar series. But I just wanted to acknowledge that we're getting some good comments in the Q&A, and it's really helpful to take those in and give to the dialogue group for further deliberations. I see, Jim, you've joined us, so you have a few comments that you'd like to share, so I'll turn to you next. Want to encourage, again, people to submit to the Q&A and have panelists also engage in that, and then I'll turn back to you, Norm, for additional questions. So Jim, over to you, and thank you for joining us.
Yeah, thank you. Sorry for being late. My government computer is so secure that even I can't get into it sometimes. It's quite amazing. Yeah, I just want to echo the comments made by Tim and Mr. Rice from California Farm Bureau. This is a really complex agricultural landscape that we're dealing with here. There's a lot of potential factors, and we really need to figure out a way to determine which ones are priorities to really go after, which ones are the low-hanging fruit per se. We all hold a piece of the truth. We all hold a piece of the answers. The question becomes is how do we populate a body of knowledge? It's not just one study. It's not just 2 studies or 10 studies. It's a body of knowledge. And that's really what we're grappling with here. And I think from Dr. Peng's presentation this morning, you can see how risk modeling can help you determine which ones will provide you with the biggest bang for the buck, which ones are going to give you the biggest payback, which areas don't you know about. So I think I just wanted to add that in is, I think the roadmap that CDFA has created is a fantastic first step. It creates a roadmap. The second thing you need to do is create a roadmap using some type of qualitative and then potentially quantitative risk assessment model, which shows you from field all the way through harvest to the consumer, where you can push, what you don't do and don't know, first of all, because you're going to have a lot of zeros in there. You're not going to know a lot of things. And then once you do learn things, which ones will make the biggest impact? And I think that science in a risk-based, science-based manner will help us figure out what we need to do. It's a long-term play, but I think it's the only way we can do this, because without it, we're all just shooting in the dark and trying to take a guess based on our personal experiences. So that's all I wanted to share. Thanks, Abby.
Thanks so much, Jim. And Michelle, Dr. J. Russell, you wanted to comment on some of the previous comments, and then Norm, I'll turn back to you.
Oh yeah, I was just going to make a comment about the two recent questions in the Q&A. Regarding the filth flies, I did make mention that E. coli 0157 has been isolated from filth flies and other fly species, both under natural conditions as well as through experiments, and they're on the list. And it's again the complexity of, who is it, that mechanism to go from these reservoirs where there are many to the actual plant that in theory is the hostile environment for E. coli 0157. So what's letting it get all the way through the supply chain when we know, especially on the Central Coast, that the E. coli 0157 occurs in the rangeland cattle, but in a very sporadic way with periodic spikes, as Rob Atwell presented. It's in wildlife, but generally at lower levels. And so population density becomes a thought. And that's the same thing with the filth flies. That's going to be on a site-by-site basis to see if that's a major problem that might need to be addressed. And regarding the vaccine, I think the vaccine is, has some promise. Right now, it's I'm not sure if Deborah saw Gabby, who's on the panel too, her presentation, kind of where we're at in terms of looking at this vaccine. It went out of production. I think it could have a niche application for the issues with proximity to livestock and as well as wildlife, and in that if it could reduce environmental loading that happens unpredictably and sporadically, it could be one hurdle against the spread of this bacteria into the produce fields. It's not a be all end all, but I would just personally say, interacting with the vaccine company, and Gabby can add, this is not a grab. This is not a money-making vaccine. There's no expectation of that from the company's side or our side. And in fact, we kind of see if this has a use, it might need to be subsidized. So I'll stop there.
Yeah, and I can add to that a little bit. Yeah, the vaccine. So yeah, I don't know if everybody saw my presentation, but the vaccine and pretty much all the brunt of the research that has focused on cattle and reduction of E. coli 0157 has really focused on feedlots or CAFOs, as they're also called. So this is really a little bit uncharted territory that we're entering with a lot of these prevention reduction measures in grazing situations. And all the prevention measures in feedlots have really been targeted towards the slaughter process, we're focusing on one time point, and that is at the end of that animal's lifespan, when it goes into the slaughter process, it becomes meat. And so it's really a shift in thinking about it when we go to a cow-calf operation or a greater stalker operation, where we really have to not think about it's that one moment in time when we're trying to reduce the risk, but it's really more of a, well, as Michelle said, the environmental load. And we all know it's all complicated. There are a lot of factors involved. There's wildlife, there are flies, there are people, the wind. I mean, we all know it's a very complex system. But yes, this vaccine, it's one of the measures that we think may be able to be applied in this situation. And may, I'm always saying may, because we don't know yet what the answer is, but may be able to reduce that environmental load. And so if that's possible, and it's, as you said, this is not about selling the vaccine, but if this really would be one way to be part of the solution rather than the problem, then I don't think we should put it aside. I think we should try and see if it really offers that opportunity. And if it does, then how it's implemented, I personally think to hopefully have that's my stance.
Thank you, Gabby. I know this will be an ongoing part of the discussion for the CAN dialogue group, but I know, Scott, you want to make a quick comment, and then I'm going to shift to Norm, because vaccines is just one thread of the bigger picture that we've all noted is complicated. But I wanted to go to Scott and then back to Norm.
I would really like to thank the CDFA and Dr. Meyer for funding the research on the E. coli vaccine. As we stand right now, to my knowledge, it is being funded by CDFA. We have not reached that level to where we don't know what the research is the outcome is. And as far as from the ranching community, I think it's my job to go back to them and say, hey, and I have been telling them, this is all research. We're funding, they're funding it. We don't know the costs, if any, that are going to come back to the ranching community at this point in time. So there again, thank you, CDFA. It's just one more level in trying to find something to help out with the entirety of food safety for our consumers.
Thanks, Scott. Norm, we've got about 15 minutes more for questions. So if you want to queue up a couple of more questions to the panelists.
Sure. Well, while we're on this particular topic, we did get a lot of questions related to this topic or this discussion. And what does current research show about the transference of STEC between wildlife and cattle, and particularly in rangeland areas? And if a herd has a high bacterial load, then does that indicate or seem to show that animals found in the same environment have that same high incidence of STEC?
I can comment on that in terms of the longitudinal studies that have already been done on the Central Coast, as well as down in the desert region. And in terms of the concentration, we have some data for cattle, and we're collecting more data with this vaccine trial in terms of, getting a handle on super shedders, so to speak. That is extremely hard to do in wildlife because the volume of sample that you get for a lot of the animals that are in the ag areas, such as birds and rodents, are not conducive to do that kind of quantitative work through culture. Or you have larger species like pigs and deer, but not access to those samples. It can be very expensive and difficult to get those samples. So we definitely have molecular data that shows that the 0157 and other STEC strains do pass back and forth between wildlife and cattle under some situations, as well as getting found in surface water. But putting all those pieces together and linking it back to a produce field is still remains a mystery to me. It's and the whole dilution factor that Trevor brought up is, as these pathogens move and they die off, it's hard to wrap your head around that. But yeah, cattle and wildlife are potential reservoirs that have to be factored in to the equation.
Great, thank you. And Trevor,
I just wanted to pile on, and Michelle probably laugh, here, but part of the problem again is to be able to, determine a lot of this source tracking, which is part of what we need to happen. You need to have, access on both sides of the fence. But I'm actually teasing her, I'm reminding her that back in 2005, going out and looking, trying to do some of this early, source tracking event and getting a lot of samples from Canada geese, and kicked myself at the time. But I was looking for, we were looking for a very specific subtype of the 0157, and almost all of the samples from Canada geese had some STEC in them, just not the right one. And they acquired it somewhere, and I won't go into any of those details, but I mean, clearly the interaction, and we know that they were, they were in proximity to types of cattle as a potential way to acquire it and sustain that within that bird population. So I mean, I'm just saying it's pretty straightforward that when there's that commingling, if you will, or visitation and feeding on cow pies and whatever, they're going to pick it up and at least retain it for a period of time.
Great, thank you. Great input. So Tim York, we have a question directly for you, and this relates to the complexity that everybody's been talking about so far this morning. And the timeline for this knowledge gain seems to be much longer than the urgency of actions needed by the leafy green sector. So do you have an area of focus that you see really as a priority for LGMA?
Well, as I mentioned earlier, we have just completed work on our adjacent land use, again, looking at the cattle in proximity to growing regions, and then the work that we've done through our committees or technical subcommittees on soil amendments. And those will be launching here in the next month or so through our partners, Western Growers, and getting those out to our growers. Those are two areas of priority, is helping growers implement those changes. It's one thing to launch them, it's another thing to equip them to put those changes into place. And then we recently announced pre-harvest testing as a requirement that'll be in place as a metric as well, so that when there is elevated risk factors, we are calling for pre-harvest risk-based testing that Dr. Suslow has helped us develop the metrics around that process. So those are three key areas that we've been working on. And as somebody said earlier, we've got to be sure that we're not just running for noise, and we're really looking where the issues potentially are. And so we're trying to be methodical about it and thoughtful about it, because quite frankly, we've made a number of changes with our growers over the last few years with the new water metrics and sanitation metrics. We've got to be sure that we're really being thoughtful about what it is we're doing, and that it's going to have some positive impacts and not just some busy work, if you will, for the growers. So we're trying to be very thoughtful about what actions we're launching, getting the support of our technical committees as well as subject matter experts to help inform that kind of work.
Great, thank you. And, kind of along this line, and not to keep you in the hot seat here, but, one of the questions that we got was that the pre-planting assessment of water quality for constituents is maybe a key factor in reducing risk, but is that really relevant when the contamination is mostly showing up closer to harvest?
Well, there's ongoing assessment that needs to be done on water. There's ongoing assessment that needs to be done on land and adjacent risk issues. So there might be factors that are not there when a crop goes into the ground, but we've got to keep our eyes open all the way through harvest to be sure that we're taking into account any additional factors that may come up. So whether it's water, or it's cattle movement, or wildlife movement, or a weather event, all of those factors have to be taken into place before we're harvesting a crop and assessing those risks. And we're equipping growers to do that with a risk assessment tool to walk them through that kind of process from pre-plant all the way through harvest.
Great, thank you. I'm going to address this all to the panelists now. Are there other adjacent land use considerations that we need to take into account here? We've been focusing a lot on rangelands. I'm just wondering if there are other land use conditions or considerations that we need to include in what we use for discussion items moving forward.
Hi there. Can I answer that question?
Yeah, as a cattleman, please do.
Well, yeah, I mean, get me away from the cattle end on this thing. Jiminy Christmas. It's interesting if you look around the Salinas Valley, specifically if you go north of Salinas, you've got a lot of suburbia around the lettuce fields and stuff. I was talking to a fella the other day. He said it's a nightmare just keeping during this whole COVID-19 deal, trying to keep people out of walking through the fields. I mean, we as a livestock producers, I said the last time, we've got 2 sets of fences. I know majority of the farmers have got fences on the river. So I don't know if I'm answering that question, but we probably need to start looking back at at our suburbia. And the other question that was brought up somewhere was our homeless. I mean, we've got a big homeless problem. I assume. I mean, you drive Highway 101, you see them. And hey, why not eat lettuce? I mean, it's a handy food. It's free. You just walk out and grab it, right? If you're homeless, you need something to eat. I mean, so maybe target that end of things.
Yeah, no, that all makes sense. And I'm just wondering if there are other land use issues that we need to consider here. All right, well, we can contemplate on that one, and I will move on to the discussion we had on biosecurity. And one of the questions we got post presentation was, how can biosecurity be maintained in conditions not conducive to full control by the farming operation? For example, employee parking alongside fields is often haphazardly done due to space. What is the priority of this particular risk or others that can't be fully controlled by the farm operation? Okay, I guess I get the prize for stumping the panel today.
Did you say something about biosecurity, Norm? I was reading
Yes,
something.
yes.
Is this dealing with the lunch wagons and stuff that come in?
Possibly, yes.
I mean, you've got your food safety people, and we're always trying to create jobs. Why not set somebody out there on the field? You go by the outhouses and stuff, the portable outhouses, and there's a sign on the back of the trailer that says, this is being watched by so-and-so. They always give you a phone number if there's a problem. I mean, not saying that leafy green guys should have one more step to do this, but maybe set a guy out there and don't allow these fellers on these ranches, because you don't know where they've come from. Most of the time, they're just driving right in off the road. Fortunately, on the rangeland side, we've got a lock gate and a good fence. So,
well, I guess what I interpreted the question as particularly relating to the parking of individual vehicles from employees. And, I think we've seen through the pandemic that busing is becoming discouraged because of health issues, and packing too many people on a bus and transporting them to a field may be an issue going forward. And I think there's going to be a number of factors that farm operations are just not able to control in their vicinity. And we're just going to have to understand what those risk factors are and how we can possibly mitigate for them.
Dr. Hack was talking about the fact that she called it the castle, I believe. I think it was Dr. Hack. She talked about the castle and the fact that we set aside areas for people to park. I know in our own operation, the vehicles are never allowed anywhere in a spot to where cattle are present. We have specific areas for them to park when they come in. And when those particular vehicles leave, specifically horse trailers, I mean, we clean up the horse manure. We don't leave it in the yard. It's cleaned up. And most of the cowboys will do that anyway. They'll pick it up. We give them a shovel. They throw it back in the trailer.
That's great. Thank you. Also should note here from Sharon Lanini that LGMA already has strict rules on vendors on farm operation visits. So appreciate that, Sharon, and that also the new water metrics are targeting assessment and treatment immediately before harvest. So good to know. Thank you for those updates there. Let's see, another question just came in here. There has been attempts to make sure that the lunch trucks that show up in the field are licensed and have met county food code and permits. That's a good point. You know, a lot of these trucks are unpermitted, and we don't really have an understanding of how they're inspected or made sure that they're using the proper sanitation there. So I think that's up to the individual farm operator to make sure they're dealing with someone who's fully permitted and inspected. Good point. Thank you, Johnny. Appreciate that.
Yeah, Norm, I just wanted to highlight the comment by Mary McGonigal Martin, who puts the human exclamation point on the charge of the California Ag Neighbor Dialogue in terms of her son being sickened and having a very serious disease from a foodborne illness, and just encouragement and support for what the CAN group is doing to press on and try to solve these issues together. I think we've got time for maybe one more question, and then we'll move to next steps for you, Norm, to lay that out and what that looks like going forward. But do you want to pick maybe one more question that we have time for the panelists to address?
Sure. So I guess this would be a pretty broad question, but hopefully we can answer it succinctly. Is there an acceptable risk level associated with wildlife transference of contaminants? And that's a broad question, so maybe that's not the shortest question to answer. And I think maybe that's what the modeling and some of the other things we've heard about here are going to lead to as a discussion moving forward. Trevor, did you have a response to that?
Too bad that I keep showing my face here, but no, I wanted to add something that maybe going forward, just to sort of change the thought process, which, you know, very much weighed into, you know, how people have addressed not so much acceptable risk, but really that's what it comes down to, but really change the conversation from continuing to look at things in terms of a percent contamination. It applies to many things in terms of risk management and processed and manufactured foods. I would argue it absolutely doesn't apply here. A 1%, a 0.5%, you know, of a population of 3 million plants per acre, or, you know, 35,000, 30,000 heads per acre is a lot of contaminated product. And so we have to drive that paradigm down to eliminating, through the practices that are in place, additional ones that might be, you know, developed or are really in development, you know, and the systems to support that, to, you know, minimize the residual risk. And then there are some downstream steps that I think we really have to focus on, you know, where at least in the fresh processed products, you know, some standards for how that's managed and how those systems are evaluated, to again minimize the risk to consumers. And so I think it's just, we have to change the conversation to really understand what we're trying to deal with, and that I think continues to be a bit of a barrier. Thanks.
Yeah, no, understood. And I think that was very well stated. I appreciate that. Thank you very much. So I think we're at the point now where we're going to kind of summarize what we've done here in the last 3 sessions now, and what we've learned. I want to thank, first of all the panelists for joining us on the roundtable today. This was great discussion, and appreciate your candid responses here today. Also to Dr. Pang and Dr. Suslow for your presentations earlier. As I said, we will make the video link available after we conclude this webinar, so that everyone can go back and review those presentations if you need to, or please advise some of your friends and neighbors who may not have seen this in person that they can review it later on too. So I think overall we've learned a lot in these 3 webinars. I know I have, and I don't profess to be a food safety expert myself, but I sure have learned an awful lot through all these subject matter experts who have presented on various different topics. So really appreciate all the input, and plus all the response that we got from the participants related to questions and also the surveys after the webinars were completed. So again, thank you all. What we're going to do at this point is our goal is to finalize a report towards the end of this year or the beginning of 2022, and really focus in on some of the practices, management efforts, and really the cross sections of where we need to focus our additional discussions moving forward. This is by no means going to be conclusive towards the end of the year, even though we're issuing a final report, so to speak. We're still going to continue these discussions moving forward, because this is probably an issue that we need to continue dialoguing on. And now that we have a great start of talking on both sides of the fence here, and involving the stakeholders as much as possible, I think we've really got a good process moving forward, and we want to take advantage of that as much as we can. We're going to convene a steering committee meeting and then a full dialogue committee meeting here in August, and continue these discussions. And hopefully we can take what we've learned here and really focus on the areas that we need to continue learning knowledge and understanding where our pressure points are, as well as where we need more information, and how will those ongoing studies, as well as the longitudinal study and the vaccine study, help us formulate some of our responses, particularly for the final report. We continue to want to further engage this community as much as possible. So that means as we come up with ideas and concepts that we need some more subject matter experts on, we are certainly going to schedule webinars as needed. So stay tuned for that. And then we also have at least 2 more town halls that we're going to plan as we move along through this process in the late summer, fall months, to really keep everyone updated on where we're going with the dialogue group, as well as the steering committee, and how this is all falling together for all of us involved here. So again, thank you for your participation today. Great conversation from all the panelists, as well as the participants with your questions. We will record those, so thank you very much. If we didn't get to them, believe me, we have recorded them, so appreciate it very much. And with that, I'm going to say thank you to Abby for facilitating our conversation here today, as well as facilitating all our other meetings. We really appreciate all your efforts. So with that, I'm going to conclude the webinar, and thank you all for your participation.